Hi there, my name is Arya. I'm a junior in Columbia College studying biochemistry and computer science, and today I'm going to talk to you a bit about my research in Dr. Peter Andolfato's lab, where I study adaptive evolution and evolutionary genetics. My research focuses on the sodium-potassium pump, which, among other things, allows for neural signal transduction and muscle contraction. It's expressed ubiquitously throughout the human body and in animal systems in general. Enzymes like the sodium-potassium pump are activated when a particular chemical with a specific molecular shape binds to it, called a substrate. Toxins work by mimicking the shape and structure of the actual enzyme substrate. Once bound to the enzyme, they hinder or even stop the function of the enzyme. One group of toxins, called cardiac glycosides, render the sodium-potassium pump entirely ineffective. It just stops working. This is detrimental, fatal. But for one group of insects, this is not true. They're able to consume plants containing cardiac glycosides or CGs without a problem, and this is interesting. We looked to the sodium-potassium pump's genome to find out why. We computationally identified two amino acid sites in the sodium-potassium pump that are clearly associated with CG resistance, 111 and 122. In order to measure the direct effects of substitutions at these sites with CG resistance, we created a set of Drosophila, or fruit flies, that contained these mutations and tested their fitness using survivorship assays. But when genetically engineered into the Drosophila model, these two substitutions in combination proved lethal. This was unexpected. Aren't these substitutions supposed to improve survivorship by making the flies insensitive to the toxin? We then noticed a third site, 119, which ameliorates the lethality caused by the presence of substitutions at sites 111 and 122. There's a great correlation between substitutions at these three sites, indicating that the substitution at site 119 is permissive. While not directly implicated in CG sensitivity, it is required for resistance. This allows us to conclude that there are a distinct set of steps required for resistance to occur. I've been testing the spectrum of insensitivity along this path of directed evolution in wild-type populations, and by analyzing interspecies CG sensitivity, we'll get a better sense of the short-term architecture of adaptation. The results from this genome-wide association study will point to the sites in the genome that cause variation in CG resistance. And eventually, we'll be able to apply this model to a wide range of adaptively evolved proteins, effectively developing a framework for adaptive evolution. Thanks so much. If you have any questions, feel free to email me at asr2210@columbia.edu. Thanks again.